Between 1347 and 1353, plague swept across Europe, North Africa, and the Middle East, killing an estimated 30 to 60% of Europe's population. Whole villages were wiped out, cities lost half their inhabitants. The social, economic, and cultural fabric of medieval civilization was torn apart. For centuries, historians knew the Black Death arrived in Europe through Mediterranean trade routes, likely via Genoese merchants sailing from the Black Sea. But where did it come from before that? Where was the source? The origin point where the pathogen that would kill tens of millions first jumped into humans? 1338, Kara Jigash, near Lake Issyk-Kul, Kyrgyzstan. On Nestorian headstones, one word repeats: pestilence. A century later, ancient Asian DNA pulled from these graves will test a 700-year argument about where the Black Death began, but nobody made the connection, not for over 100 years. In 1886, when Russian archaeologists excavated the cemeteries at Kara-Jigak, they meticulously recorded what they found: grave locations, headstone dates, inscriptions, skeletal positions. It was professional work, thorough for its time. But the field notes were filed away with the bones and stones, gathering dust in museum archives. The gravestones themselves were curious. Dated 1338 and 1339, a tight cluster. Medieval cemeteries usually show burials spread over decades, sometimes centuries. But here, the vast majority were concentrated in just 2 years. That should have raised questions. But without context, without a framework to interpret the pattern, it was just data. The skeletal remains sat in storage for over a century, bones in boxes, waiting for someone to ask the right questions. That someone was Philip Slavin, a medieval historian who specializes in pandemics, climate, and economic disruption in the 14th century. In the 2010s, while researching early plague outbreaks, he came across references to the Kara-Jigach excavation. Curious, he tracked down the original field notes, the 19th century documents that had been filed away and largely forgotten. And when he went through the burial records carefully, counting dates, mapping the chronology, he saw something extraordinary. A massive, concentrated surge of deaths. 118 burials in just 2 years. In a small community, that's catastrophic. That's not normal mortality. That's a crisis. Slavin suspected he was looking at an early plague outbreak. But suspicion isn't proof. To confirm it, he needed evidence that Yersinia pestis, the bacterium that causes plague, was actually present in those bodies. He needed ancient DNA. And just like Slavin needed DNA, we need your like and subscribe to keep these documentaries going and to make them better for you every day. Thank you for being here. Slavin reached out to researchers at the Max Planck Institute for Evolutionary Anthropology in Germany, a world leader in paleogenetics. For years, the Max Planck team had been building a global family tree of Yersinia pestis, sampling modern and ancient strains from over 1,300 sources, mapping how the bacterium evolved and spread across continents and centuries. And they had already found something intriguing: a diversification event. A moment in the pathogen's history when a single ancestral lineage split into 4 major branches almost simultaneously. Geneticists call this a Big Bang diversification. It happens when a population explodes, when conditions allow rapid expansion and branching. And this Big Bang happened just before the European pandemic, right before the Black Death swept into Europe. The Max Planck team suspected they were looking at the genetic signature of the Black Death's origin, but they didn't know where that origin was. They didn't have a geographic anchor. They needed an ancient sample from the right time and place to pin down the source. Philip Slavin's Kara-Jigach skeletons, dated 1339, were exactly what they needed. If those bodies contained plague DNA, and if that DNA matched the ancestral lineage at the root of the diversification, it would prove the Black Death started in Central Asia. But could they extract bacterial DNA from 700-year-old bones, and if so, would it be the right strain? The team requested permission to sample teeth from 7 Kara-Jigach individuals. Teeth are ideal for ancient pathogen recovery because the dental pulp, the soft tissue inside the tooth, can trap blood-borne bacteria during infection and preserve them for centuries. The hard enamel shell protects the interior from environmental contamination. The sampling process is delicate. You can't just grind up an entire tooth. You drill carefully into the pulp chamber, extract powdered material, and hope the bacterial DNA is present and intact enough to sequence. Out of 7 individuals sampled, 3 yielded unmistakable Yersinia pestis DNA. 3 confirmed plague victims from 1338 and 1339, 7 years before the Black Death arrived in Europe. But finding plague DNA in Central Asia wasn't enough. Plague is endemic to parts of Asia. It circulates in wild rodent populations. Finding it in medieval remains could just mean local cases, unconnected to the European pandemic. To prove the connection, the researchers needed to sequence the full bacterial genome and compare it to the global family tree they'd been building. Painstakingly reconstructing ancient genomes from degraded, fragmented DNA, they assembled complete Yersinia pestis sequences from the Kara-Jigach samples. And when they placed those genomes on the family tree, the result was unambiguous. The Kara-Jigach strain sat at the root of the diversification, right at the base of the 4-branch Big Bang that preceded the European pandemic. This was it, the ancestral source, the lineage that gave rise to the Black Death. But genetics alone doesn't explain how or why plague jumped from animals into humans at that specific time and place. To understand that, you need ecology. The bacterium, Yersinia pestis, lives in wild rodent populations, marmots, great gerbils, voles, and other burrowing mammals. Fleas feed on infected rodents, pick up the bacteria, and transmit it to other animals. The system is stable, endemic, cycling through wildlife populations without necessarily spilling over into humans. But ecological balance is fragile. A shift in climate, a change in vegetation, a population boom in rodents, any of these can tip the system towards spillover. In the early 14th century, evidence suggests just such a shift occurred. Climate reconstructions indicate wetter conditions in parts of Central Asia during this period. More rainfall means more vegetation. More vegetation supports larger rodent populations. Larger rodent populations mean more fleas. More fleas mean higher transmission rates. When rodent populations surge, they expand into new areas, including places where humans live and work. And when humans interact with infected animals, through hunting, handling, or simply being in close proximity, the pathogen can jump species. All it takes is one spillover event, one person bitten by an infected flea, one hunter skinning a diseased marmot, and suddenly, a bacterium that lived in wild rodents is circulating in a human community. That's what happened at Kara-Jigach, a zoonotic spillover. Plague jumped from local wildlife into the human population, triggering a mortality crisis that killed over 100 people in 2 years. In a small, isolated community, the outbreak might have burned itself out. But Kara-Jigach wasn't isolated. It was on the Silk Road. From the moment plague entered the human population at Kara-Jigach, the pathogen had access to one of history's greatest transmission networks. The Silk Road wasn't a single route. It was a web of trade paths connecting China, Central Asia, Persia, and Europe. Caravans moved constantly, carrying silk, spices, porcelain, precious metals, and a host of other goods. And they carried something else: rats, or more precisely, the fleas on rats. Commensal rodents, species that live in close association with humans, infesting grain stores, hitching rides on cargo, breeding in the corners of merchant warehouses. Infected fleas on rats moving with trade caravans could carry plague westward, step by step, from one trading post to the next. Each stop a potential new outbreak. Each outbreak a new opportunity for the bacterium to spread further. The transmission path from Central Asia to Europe took years. Plague didn't leap instantly from Kyrgyzstan to Italy. It moved slowly, following trade routes, hopping from city to city, outbreak to outbreak. By 1346, it reached Kaffa, a Genoese trading colony on the Crimean Peninsula in the Black Sea. Kaffa was under siege by a Mongol army when plague struck the besieging forces. According to contemporary chronicles, the Mongols catapulted infected corpses over the city walls, an early form of biological warfare. Whether that story is true or embellished, the result is clear. And from Kaffa, Genoese merchants, fleeing the outbreak and the siege, sailed for home. They carried the disease with them. Genoese ships docked at Constantinople, at ports in Greece and Italy, at Marseille in France. Everywhere they landed, plague followed. Mediterranean trade networks, dense and interconnected, allowed explosive spread. City to city, port to port. By 1347, plague was in Sicily and southern Italy. By 1348, it had reached France, Spain, and England. By 1349, Germany and Scandinavia. By 1353, it had swept across nearly all of Europe, from Portugal to Russia. The mortality was staggering. Estimates vary, but scholarly consensus suggests Europe lost between 30 and 60% of its population. Some regions were hit harder. Some cities lost 2/3 of their inhabitants. Villages were abandoned. Fields went untilled. Entire communities vanished. The social and economic consequences reshaped European civilization. Labor shortages drove up wages. The power of feudal lords weakened as peasants gained leverage. The church, unable to stop the plague despite prayers and processions, saw its authority questioned. Art and literature became obsessed with death, decay, memento mori. The Black Death didn't just kill people, it transformed societies. And all of it traced back to a small cemetery in Central Asia, where headstones marked with the word pestilence recorded a local tragedy that became a global catastrophe. If this one discovery changed how we tell the story of the Black Death, imagine an hour of discoveries just like it. Click here to start the 1-hour DNA revelations.